All right, hi everyone. I hope you guys are doing well. So in this video, we are going to briefly cover chapter 2, and we're also going to start covering chapter 3 as well. So I'm sure I'll discuss this more in the actual lecture part coming up, but I had to choose how to handle all the material we could possibly cover in this class. And I decided that I wanted to make sure we had as much time as possible to talk about specific disorders. Because of that, I'm not going to be covering everything that your textbook has in Chapter 2. Chapter 2 is going to be about research methods, and my goal here is to give you enough information to be able to understand the studies that we're going to be talking about throughout the semester without spending so much time on research methods that it takes away from the actual content of the course, specifically abnormal psychology. So we will briefly discuss research methods, and then we will also start Chapter 3. Chapter 3 is going to talk about ways that modern individuals today think about psychological problems. Where do they come from and how should they be treated? So we'll start Chapter 3 in this video as well and then wrap up Chapter 3 in the next video. We will talk about the topic for Activity 2. Remember, for these, I'm looking for your original work, and I'm looking for a response that is at least 200 words long. Make sure that if you do use any sources that you cite those appropriately. Let me know if you have questions. Let's go ahead and get into lecture.
All right, so today's lecture is going to start off with a crash course in research methods. So this is not a research methods class. However, you have to understand certain terms. When we're talking about different, for example, treatments later in the semester, you need to understand how we decided these treatments were effective. How do we know that they work? What kinds of research is done in the field of clinical psychology? So this class is especially challenging for me to teach in a unique way because there's so much information that I want to communicate that I have to choose where to spend my time. So what I want to do is give you enough information about research methods to be able to understand the other information in the class, but not so much information in research methods that we lose time that could be spent more towards abnormal psychology, which is obviously the focus of this class. So this first part of the lecture is just intended to help you understand some of the terms that are going to come up again later in the class. It is by no means a comprehensive research methods class. However, I feel confident that you guys will be getting comprehensive research methods classes later. All right, so when we're talking about research, there are many different types of research. The three most common would be observational research, correlational research, and experimental research. Now, what we want to understand here is that it's not a situation where one type of research is better than another type of research per se. It's more that the different types of research have different goals. So I have these listed here. Observational research has the goal of description, so it allows us to describe what's going on. Correlational research allows for prediction, which means that if we have some correlational information, then we can make predictions about the future, not necessarily perfect predictions. We'll get to that in a second. And then finally, experimental research is going to answer causal questions, basically help us understand why something is happening. So these different types of research are all important for their different methods are important for understanding different kinds of problems. However, what we do want to know is that with experimental research being the only kind of research that allows us to determine causation, we need to be aware of that when we're reading research studies. So sometimes researchers will do observational research or correlational research or some other type, and they will write up their results in a way that can be confusing, that can make it sound like they have done experimental research. So what I want you to be able to do after watching this lecture video is to be able to understand what actually makes something an experiment and to be able to tell the difference between experimental research and other types of research. So starting off with observational research basically just means that we are studying a phenomenon that's happening naturally and we're making observations about it. Obviously that's right there in the name. Now, with observational research, we are not manipulating anything, we're not controlling for anything, we're just watching something happen and making notes about it and describing it. And there are several different ways to do this. I listed a few examples. Probably the first one that comes to mind would be naturalistic observation. So when we talk about observational research, perhaps you think about going to a park. If you're studying parenting, maybe you go to a park and you watch the way parents interact with their children. Or maybe if you're interested in maybe bullying, you go to a school and you observe how kids behave in the classroom, how they treat each other, that kind of thing. That's naturalistic observation. But that's not the only type of observational research. So archival research is also listed here. An archive is just basically documents that we have saved. So it would still technically be observational research if you, for example, did a study on standardized tests, and maybe you looked at test scores over time. Maybe you could look at things like birth certificates, death certificates, marriage certificates, that kind of thing, looking at documents that have been saved. You're still making observations here, and you're still doing this for the purpose of description. And then finally, with a case study, usually the typical case study would be one person. It could be possibly 2 or 3 people, but a very small sample size. And a case study looks at a particular issue, a particular person perhaps, in depth. So the classic example of a case study is Phineas Gage. I don't know if you guys have heard of our friend Phineas Gage that we like to talk about in psychology classes. Phineas Gage worked on a railway. It's sometime in the 1800s, I believe, and there was an explosion, and a railroad spike went in under his chin and came out through the top of his skull, and amazingly he survived. So, as you can imagine, people were interested in how this might impact a person living with that kind of significant trauma and damage. So case studies would be a perfect way to study that kind of situation. With a case study, you can't get a large sample size, maybe because that kind of thing is very rare. It's not like we have a lot of individuals who are suffering that particular type of trauma. But with a case study, you get a lot of in-depth information. So if I had Phineas Gage in front of me, then I would be interested in doing medical testing. I'd be interested in interviewing him, interviewing family members, interviewing friends, to see how things maybe change before and afterwards, maybe do some testing, some intelligence testing, personality testing. So a case study is basically getting a lot of information on one particular person. So these are just a few examples of observational research. The benefits of observational research, because it does allow for description, it's a really great way of getting ideas. Maybe ideas for future correlational or experimental research studies. When you think about naturalistic observation, being able to watch someone in their natural environment gives you a lot of really great information. Sometimes people will behave very differently when they're in the lab or when they know that they're being part of an experiment, but just naturally watching people in their own environment can give you some really great information. Unfortunately, with every type of research there will be some drawbacks associated. So the thing about naturalistic observation in particular is that we call it reactivity. The idea that if a person is aware that you're watching them, it will change the way they behave. And that's kind of a normal human response. If you notice that someone is watching you, following you around with a clipboard making notes, you're going to change the way you behave. So then we have to find ways to be subtle so that we don't actually change the way our participants are behaving. Also, obviously, if you're going to be doing naturalistic observation in, like, for example, a school setting or that kind of thing, you have to get permission from, at least from maybe principal, teacher, parents of the students perhaps. So sometimes there's a lot of paperwork involved with that. But other drawbacks here would be that although you get a good description, you don't necessarily understand why that's going on. So maybe you go to the park and you watch parents interact with their kids. You can make notes about how the parents interact with their kids, but you don't really know why they're interacting that way. There's a lot of information you don't get this way, and so it could potentially be misleading because you don't know what else is going on and there's not really a way for you to get that information. Also, it doesn't always generalize, and you're going to hear these terms coming up again later on in this set of slides, but for example, with your case studies, if you have one particular person who has this result, it doesn't necessarily mean that the next person you get to be part of your case study would have the same result. It doesn't mean that every person, for example, who had a similar kind of trauma to Phineas Gage would have the same outcomes. So sometimes observational research could be misleading. So just bearing that in mind. Correlational research is the second type of research we're going to talk about. When we're talking about correlations, we're talking about two or more variables that are co-related, so they go together. So for example, maybe we're interested in studying the number of Facebook friends you have and the time you spend studying for a test. That kind of research has been done. What we do is we take two variables that are already existing and we don't manipulate them at all. I don't manipulate the number of Facebook friends you have. I don't manipulate the amount of time you spend studying. All I do is collect that information and look to see if there's a relationship. So research has shown a correlation between Facebook friends and time spent studying. For example, maybe a professor is interested in the correlation between class attendance and grades. So I don't measure, I do measure, but I don't manipulate your class attendance. I don't manipulate your grade. I measure your class attendance, I measure your grade, and then I look for the relationship between them. So with correlational research, we take two naturally occurring variables and just look to see if there's a connection. Now, a couple of things you need to understand about correlations. First of all, the direction, and then we're also going to talk about the strength. So when we talk about the direction of a correlation, we're talking about, is this a positive correlation or a negative correlation? And really that just means, are they moving in the same direction or in opposite directions? So with a positive correlation, we have two naturally occurring variables that increase and or decrease together. So for example, class attendance goes up, grades go up. Class attendance goes down, grades go down. So that's a positive correlation. They're moving in the same direction. The amount of donuts I eat goes up, weight goes up. Donuts I eat goes down, weight goes down. I mean, you can think of a lot of examples here of situations where two variables go up together or they go down together. On the other hand, a negative correlation means that as one variable goes up, the other variable goes down. So we're still measuring two things that are naturally occurring. We're not manipulating them, we're just measuring them. But with a negative correlation, they move in opposite directions. So as exercise goes up, weight goes down. Exercise goes down, weight goes up. We could say absences go up, grades go down. Absences go down, grades go up. So they're moving in opposite directions here. Still strongly related to each other, just moving in opposite directions. Just for your own personal information, there's research to suggest that as Facebook friends goes up, time spent studying goes down. Facebook friends goes down, time spent studying goes up. The idea behind that is probably that the amount of time that you spend on social media is probably less time that you're spending studying. So that would be a negative correlation. All right, so as always, let me know if you have any questions about these terms. So we need to know the direction of a correlation. To say that two variables are correlated doesn't tell us a whole lot without knowing the direction. But we also need to know the strength, because the strength tells us how strongly they go together. Do they go together all the time or only on occasion? So the stronger the strength, the higher the strength, then the better the prediction. So correlational research is allowing us to make a prediction. So if I know the correlation between class attendance and grades, then I can take a student's attendance and predict their grade. Or I can take their grade and predict their attendance. This is intended to be helpful, but it's only helpful if we have correlations of a certain strength. So the strength is going to be represented by a number between 0 and 1. Correlation can be anywhere between 0 and 1. However, the closer the number is to 1, the stronger the correlation. So a 1.00 correlation is a perfect correlation. It means that those two variables always go together in the same way. This is very rare. It's very rare to find two things that always go together. On the other hand, a correlation of 0 means that these two things don't go together at all, like they're never associated with each other. And once again, that's pretty rare as well. It's kind of hard to find two things that are not related to each other even a little bit. So mostly we'll have numbers in between 0 and 1. The closer the number is to 1, the stronger the correlation. So, for example, a 0.5 correlation is stronger than a 0.3 correlation. Why? Because 0.5 is closer to 1. A 0.7 correlation is stronger than a 0.6 correlation because 0.7 is closer to 1. Now, when we're talking about the strength, we're only talking about the number. So I have some examples down here for you. One thing to remember is that the direction does not impact the strength. So the direction refers to whether we have positive or negative correlations. That is important information, but it does not impact our strength. So a 0.9 correlation is stronger than 0.6 because a 0.9 correlation is closer to 1. It's a bigger number. A negative 0.9 correlation is stronger than a positive 0.6 correlation. Why? Because 0.9 is a bigger number. It's closer to 1. A positive 0.9 correlation is stronger than a negative 0.6 correlation. Why? Because 0.9 is a higher number. It's closer to 1. So when you're thinking about the strength, you only look at the number. The direction does not tell you anything about the strength. The direction tells you, are we moving together in the same direction or are we moving in opposite directions? That's important information. But it does not tell you anything about the strength. So if there's a test question, for example, that asks you, which of the following correlations is stronger, negative 0.7 or positive 0.7? One of the answer choices would be neither, they have the same strength. And that would be the answer you would be looking for. A negative 0.7 correlation has the same strength as a positive 0.7 correlation. The number tells you the strength. Okay? Let me know if you have any questions. All right. Now, we have already said that experimental research is the only kind of research that allows us to determine causation. Correlation does not equal causation. So, if I do a correlational research and I measure your class attendance and your grades, even if I find a perfect correlation, which is very rare, even if I find a perfect correlation, I still cannot say that your class attendance caused your grades. That might seem like an obvious leap. That might make sense to you, but I can't say that because this kind of research does not allow for that. Because I didn't manipulate anything, I didn't control for anything, I just measured naturally occurring events. And there is always the possibility that a third variable caused both of those things, maybe caused both your class attendance and your grade. So when you're talking about correlations, you need to remember that correlations allow for prediction. They do not allow for causation. The classic example that's typically used here has to do with violent crime and ice cream sales. So there is a strong positive correlation between violent crime and ice cream sales. Let that soak in for a second. As violent crime rates go up, ice cream sales go up. Violent crime rates go down, ice cream sales go down. Now, let's think about that. Is it possible that violent ice cream sales violent ice cream sales. I guess that's possible. Is it possible that violent crime causes the ice cream sales? Like I have the munchies after that violent crime. Or is it possible that the ice cream sales caused the violent crime? I kind of wondered a while back when Bluebell went out for a while, and when Bluebell came back, I kind of thought Kroger would be mobbed of people trying to get Dutch chocolate. But that's a whole other conversation. No, I can't say that. Is it possible that there's a third variable causing both? And this is where I make my PMS joke that I'm allowed to make because I'm a woman. Guys, I don't recommend that you make that joke. But no, is there a possible third variable, something that could be causing violent crime to happen and something that could be causing ice cream sales to go up? And the answer is the temperature. Violent crime happens more during the summer because if you take social psychology, you'll see that anything that makes you uncomfortable makes you more likely to be aggressive. Also, maybe more people out to be maybe getting on your nerves. I don't know. On the other hand, ice cream sales are also higher during the summer. There's not really a wrong time to eat ice cream, but when it's hot outside, people tend to eat more ice cream. So just because those two things are strongly correlated does not mean one caused the other. There's always the possibility of a third variable causing both. So if that's the first time you've heard that, you'll hear it again, I'm sure. Correlation does not equal causation. Okay, so if we want to understand causation, then we're going to need to do experimental research. And of course we want to understand causation. The whole reason we go into psychology is to understand why things happen, right? Now, experimental research, because it does tell us something that other types of research don't tell us, is also going to be more complicated and going to require more of us. So there are several different terms on this slide that are important to understand if you're going to understand experimental research. So first of all, experimental research, allowing us to determine causation, is going to require us to start off by identifying our population. Your population would be all of the people that you're interested in studying. So let's say, for example, that we have a new treatment for depression. Our population would be all individuals with symptoms of depression. Now, the question is, can I get every person with symptoms of depression to be in my research study? And the answer to that is obviously no. Because, first of all, the sheer number of people, there's no way we could get that many people to be part of our research study. Also, we can't force people to participate who don't want to participate. That's not ethical. There may be some people who have symptoms of depression, but they have never been diagnosed, and so we don't have any documentation or any way of knowing about that to reach out to those people. So no, there is no way to get your entire population to be part of your research study. We would love to do that, but unfortunately we can't. So what we're going to do is to take a sample. A sample is going to be a selection of people from your population who will actually take part in the study. So while your sample may be thousands and thousands of people, you're going to have a sample, and the sample size depends on how much money do you have, how much time do you have, how many research associates do you have. The larger the sample, typically the better, because a larger sample would reflect your population better. So for example, if we have a population of 2,000 people, we really need a big sample. I mean, 200, 300 people would be fantastic, although we're not always able to get that many. So the population is everyone you're interested in. The sample would be the subset of the population that actually takes part in your research study. Now, how do you choose your sample? Well, the best way to choose your sample is to use something called random sampling. What this means is that every person in the population has an equal chance of being chosen to be part of your research study. Now, if you have a small population, this is really easy. So, for example, if abnormal psychology is my population, if I'm interested in you guys, and there's 25 of you, and I randomly select a sample of 5 people, it would be really easy for me to take 25 names, put them in a hat, draw out 5, and that's my sample. Unfortunately, when your population is huge, like all of the people in North America with symptoms of depression, it can be very hard to get a random sample. However, the better job you do with your sample, the better your research. So you want a large sample, and you want a sample that looks like the population. When you randomly select a sample, it's more likely to look like the population. And when I say look like the population, I mean you want to have the right percentage of different ethnicities, different socioeconomic statuses, different education, different family background, that kind of thing. Anything that you think could be relevant to your research study. So this is not easy to do, but we do the best we can here. We get a random sample. Now, once you have your random sample from your population, then what you're going to do is randomly assign them into groups. So, with my depression example, let's say that I'm interested in this new treatment for depression. My population is everyone who has symptoms of depression. I cannot get that many, so I randomly select a sample. Let's say I select 100 people. That's my sample. Now, once I have randomly selected my 100 people, I randomly assign them into 2 groups. We'll just talk about some simple research designs here. 2 groups. Now, what that means is that there'll be 50 people in each group. Now, the typical example here would include a control group and an experimental group. So I randomly assign people, half of them into my control group, half of them into my experimental group. Now, the experimental group is going to be the group that's going to actually receive my new treatment, whether that be a medication or a therapy. They're going to get the actual treatment. The control group is not going to get any treatment at all, or they'll get a placebo. A placebo is basically a fake treatment where a person, for example, maybe takes a pill that doesn't actually have any active ingredients. There are also placebo therapies as well, as strange as that sounds, a therapy that doesn't actually involve anything proven to help someone, but that's another conversation for another class. But The control group does not get the new treatment, basically, and the experimental group does. Now, why do we have to have a control group? Why not give this treatment to everyone? Well, if I give my treatment to everyone and everyone gets better, I don't know that it was my treatment that made them get better. What if there was just this fantastic change in the weather, and the weather is just really beautiful, and so everybody felt a little bit better because of that? There could be any number of things that could cause people to feel a little bit less depressed. If I have a control group, then I can compare, and I would expect, if my treatment is effective, that my experimental group would have more of a benefit than my control group. Although the control group would probably get better at least a little bit, because being part of a treatment gives people hope. The placebo effect is real. If you give someone a pill and convince them that it does a certain thing, then they will have that outcome, at least to a certain extent. However, our experimental group should have a greater outcome, greater benefits, assuming that we have a good treatment. Now, this leads us to the terms independent variable and dependent variable. This is what makes experimental research experimental. The independent variable is the thing that you manipulate. It's what you control. It's really what you controlled for. So in this research study, the thing that I manipulated as the researcher is the type of treatment a person received. In the experimental group, they received a real treatment. In the control group, they received a placebo fake treatment. So the independent variable, the thing I manipulated, is the treatment in this particular example, which means then that if I do a good job, I can say my treatment caused my outcome because I actually did manipulate something between the two groups. The dependent variable is the variable that we measure. So, for example, in this example, it would be symptoms of depression. Your dependent variable is your outcome measure. So we would expect that our dependent variable would show us fewer symptoms of depression in the experimental group and more symptoms of depression in the control group. So we manipulate the independent variable and we measure the dependent variable. Now here's the question. Why do the other kinds of research? Experimental research is the only kind of research that allows us to determine causation. Why do we even bother with observational research or correlational research? Well, the answer to that, there are a few answers to that. One potential answer is that we are not always able to do experiments. Sometimes we're not able to do experiments because we can't get a good random sample, or because we're studying something that we can't control for, and all we can really do is measure it. We really can't manipulate it. Sometimes this is because there are ethical issues involved. So let's say, for example, that you want to study the effects of cigarette smoking on your grades. You can't take a random group of people and assign some people into a smoking condition and some people into a non-smoking condition. It's not ethical for you to take people who don't smoke and force them to smoke because of your research study. And it's not ethical for you to take people who already do smoke and tell them they can't smoke anymore because they got randomly assigned into the non-smoking condition. Think about why we don't know a whole lot about medications during pregnancy. Because we can't take a random sample of pregnant women and expose them to something that could be potentially harmful. So all we can really do there is correlational research because we can't intentionally put people in harm's way. So experimental research sometimes is not done because it's not ethical to do it, and sometimes it's not done because it's not practical. We don't have enough money. We can't get a good random sample. But if you can do experimental research, that's great. Now, it's important for us to do high quality research. I really can't say that enough. Why would you waste your own time and your own funding and your participants' time and the reputation of psychology on doing poor experiments? You shouldn't do that. We always want to make sure we have high quality research, because outside of just wasting a lot of time and money, what happens if you don't do a good job and your results are not accurate, but people think they're accurate and make decisions based on it? So there have been examples in recent history of people who have released information that is either poorly done or maybe falsified, and then people believe it though, and they actually make changes in healthcare and other things because they believe this research. So you can harm people with doing low quality research. So we want to make sure our research is high quality. How do we do this? Well, we need to understand a couple of terms here: reliability versus validity. Reliability refers to consistency. So, for example, if I take an intelligence test and it says my intelligence score is 105, and I take it again next month and it's 105, and I take it again next month and it's 105, that would be reliable. And it doesn't necessarily have to be exactly the same. Maybe 1 month it's 104 and 1 month it's 106, but they're pretty much clustered together. Those results are reliable. Now, obviously, in almost all cases, we want reliability in our research. But reliability is not enough, typically. So think about that. Imagine if a person took an intelligence test every month and they scored right around that 105 mark, but their true level of intelligence was 150. But the test was telling them consistently, reliably, that their true level of intelligence was 105. It's reliable, but it's not valid. So validity refers to measuring what we're supposed to be measuring. It refers to accuracy here. So we would want, for example, our intelligence test to be both valid and reliable. So, can you have reliability without validity? Yes, you absolutely can. You can be reliably wrong. You can step on a scale and have it say you weigh 1,000 pounds, and you step back off and you step back on the scale and it says you weigh 1,000 pounds. You can do that all day long, but if you don't weigh 1,000 pounds, the reliability without validity doesn't tell you anything. Now, the next question: is it possible to have validity without reliability? It is possible, but not likely. It depends on what you're measuring. So what we're saying here, is there a situation where your results are accurately measuring what they're supposed to be measuring, but they're not consistent at all? This is only possible if you're measuring something that's not consistent. But there are some things that are not consistent, like, for example, your mood. If I bring you in on Friday and you're having a great day and your mood is fantastic, and I bring you back in on Tuesday and your mood is terrible and you're just really sad that day, that doesn't mean there's something wrong with my measure. My measure could be valid. It could be accurately measuring your mood, but because mood is not reliable, it's not consistent over time, then I might not find reliability, but I could still find validity. That's rare. A lot of things we measure in psychology don't change rapidly like that, but that's one example of how that could happen. Now, there are many different types of reliability, many different types of validity. We're going to talk about one specific example here because it relates highly to abnormal psychology research. And this is internal validity versus external validity. So validity in general means measuring what you're supposed to be measuring. But there are different types of validity. Internal validity refers to making sure that your independent variable is the only thing that impacts your dependent variable. So, with our example of the depression treatment, I want my treatment to be the only thing that impacts people's level of depression. If I have high internal validity, that means there's not a whole lot of other random things impacting my outcome. My treatment, my independent variable, is the only thing impacting my outcome. My clients are not getting better because of the weather or because they hit the jackpot or because of ice cream or anything else, right? Internal validity is great. On the other hand, we have external validity. External validity refers to, are these results measuring what they're supposed to be measuring beyond my study? So, the results that I find with my 100 people who have depression, do they actually tell me something about my population? All the people that have depression. Does it actually tell me something about people who are not part of a research study, but people who are out in the real world? So external validity refers to generalization. Are my results true outside of the lab with other people, not just my sample? Of course I care about my sample, but what I really want to know is about people who have depression in general. I really want to know about my entire population. Now, these two things don't always play nicely with each other, so think about this for a second. In order to have internal validity, we have to tightly control for everything to make sure nothing else impacts our results. But what happens if you tightly control for everything in your research study? It harms your external validity, because out in the real world with real people, things are messy and they're not tightly controlled for. So just because you found something is true with a particular group of people in a lab setting that they've been sequestered and there's nothing in the world that's impacted them for the last 3 months because we haven't let them out of their hotel room, or whatever the case may be. That's not really realistic for most people. So on the other hand, though, with external validity, you say, okay, well, I want my results to generalize, so let's try to do this in a way that reflects real life. And that doesn't always include tight internal validity. So it seems like they're always at odds with each other. This is important because it leads us to empirically supported treatments. So this is abnormal psychology. We're going to talk about a lot of different diagnoses. We're going to talk about the research that's been done on various treatments. The vast majority of the treatments you're going to find have been researched using empirically supported treatments. This means basically that we tightly control for everything. We bring in individuals and we give them our treatment. But, for example, they're only allowed to come in contact with highly trained professionals. So usually the one in an empirically supported treatment, the person who designed the treatment is the one giving the treatment. So they're not going to mess it up. This is something that they know very well. Or people who have worked with them and been trained directly under them will be the ones doing the treatment. And because I want to make sure that it's my treatment that causes my outcome, I need to try to get rid of anything that could potentially impact my results. So let's don't include anyone who has substance use, because substance use could change their symptoms. Let's don't include anyone who has another diagnosis. So comorbidity refers to having more than one diagnosis at a time, and it's actually fairly common. But what we're saying here is if I have a new treatment for depression, I want to do my research having the person who designed the treatment give the treatment, and then exclude anyone who has anxiety or substance or psychotic symptoms or anything else, and that way we can be really, really sure it was our treatment that caused our outcome and not some other random variable. Well, this is high on internal validity. Tightly control for everything, so then we know our treatment caused our result, and that's great. We need to do this kind of research. It's crucial because otherwise we might be giving treatments that are harmful. Here's the problem. Out in the real world, if you're a person with depression and you walk into a clinic to get treatment, you're probably not going to get the treatment from the person who designed it, right? You're probably not going to get it from a person who's been trained by that individual. So the question is, your empirically supported treatment, will it work even if someone who didn't design it and maybe doesn't know it super well does it? Maybe this person has some training in it but not as much training. Will it still work? Will it still work if the person who walks into your clinic has anxiety disorders as well, or has substance use as well? That's the question. So we need to do research on treatments, but what we also need to understand is that just because a treatment worked with a highly trained professional conducting a treatment on patients who have nothing else going on in their lives does not necessarily mean it will work out in the real world with people who don't have as much training leading it and giving it to people who have other issues as well. So I want you guys to keep in mind that sometimes clinicians have to tailor their treatments. They have to change things a little bit to make it appropriate for their clients, for their particular situations. All right. That is the end of the research methods portion. I'm about to start the next section of today's lecture. All right, so in addition to the research methods slides for today, we're also going to start the slides from chapter 3, which is about models of abnormality. So there are many different potential models that we could talk about when we're thinking about abnormal psychology. And we are going to talk about several. My plan today is kind of generally to introduce what I mean when I say models of abnormality, and then to talk primarily about the biological model. And then during the next lecture video, we'll get into some more psychogenic models of abnormality. So what do I mean when I say model? When we're talking about abnormal psychology, the models that we use, it says they spell out basic assumptions, give order, set guidelines. Basically, your model is your understanding of, if we're talking about abnormal psychology, where do problems come from? What is the best way to treat a problem? Your model may also guide you as far as how do I know my treatment has been successful? What kinds of outcomes am I hoping for? So models influence what we observe, what we ask, what information we seek, how we interpret this information. So we briefly touched on this during the last lecture video when we talked about how we have like humanistic psychology, about how we talked about behaviorism and a cognitive viewpoint and a psychodynamic viewpoint. Your model is your understanding of why we have problems and how we should treat them, basically. So, if there's a client who has generalized anxiety disorder, they're going to be viewed differently, they're going to be interviewed differently, they're going to be given different kinds of tests, and probably going to have different kinds of treatment depending on the model of the psychologist that they go to. So always a good idea, if you're going to be seeing a psychologist, ask the person, what is your understanding of where problems come from? Are you more behaviorist? Are you more cognitive? Ask the person their model, because that's going to be important information for you to have. And if you're going to be a psychologist or counselor one day, then you're going to have to develop your own model, your own understanding of where problems come from. So, until recently, this has not been as much of an issue. So it talks here about how clinical psychologists, clinical scientists, people doing research have typically had like one prevailing model at a time. Think back to our history lecture. We talked about how, for example, there was a time when people tended to think that psychological problems came from evil spirits. And so during that time period, the vast majority of people who were working with individuals having problems would say this is a spirit issue. And then we move into a time period where people tended to think that maybe it was the different humors, maybe there was a biological cause here. And the majority of people would say there's a biological cause here, too much blood, not enough bile, that kind of thing. Today, we don't have a prevailing, norm as far as what we think of as abnormality. There are several different models today, which I think is good. I think it's a little bit short-sighted for us all to agree, based on our cultural norms, this is where problems come from. I think having different people with different experiences and different perspectives all working on the same problem is healthy and a good thing. However, having said that, it does create a little bit of conflict. So, as we go through and talk about the different disorders, there may be some pretty drastic differences in the ways that we understand disorders depending on our theoretical orientation. And your understanding of where a disorder comes from will, of course, impact your treatment. What you're going to see, though, is that no single model represents every disorder. There will be some disorders where one particular model seems to have that pretty much figured out, and then the next lecture video we'll talk about a different set of disorders, and there may be no answer for that based on the previous models. So we don't have it all figured out, and a lot of people who practice use an eclectic approach, so they have maybe been influenced by several different perspectives. But today, as I said, we're going to focus on the biological model. Now, the biological model is going to adopt a medical perspective. So the rest of today's lecture is going to have a lot of biological terminology in it. You're going to be thinking, this sounds a little bit like we're trying to treat a medical condition, and that's because individuals who hold this model would say that psychological problems are medical problems. So the main focus here is looking for an illness, viewing psychological problems as being similar to other medical issues, physical illnesses brought on by dysfunction in the body. So when a person has the flu, or when a person has cancer, we would say there's a dysfunction in the body. Well, the biological model would say that, generalized anxiety disorder and depression and schizophrenia, they happen because there's a dysfunction somewhere in the body, which typically we look to the brain, although there could be other parts of the body involved, but typically looking to the brain, potentially brain anatomy or brain chemistry. So talking about brain anatomy briefly here, This is not a biological psychology course, but we know that certain parts of the brain are connected to certain psychological processes. So there are parts of the brain that are focused on memory. There are parts of the brain that are focused on different sensory information, processing, vision, that kind of thing. There are parts of the brain that are associated with your personality and associated with decision making and attention. And so, if, for example, you have a biological viewpoint, you might look for some kind of change in brain anatomy that could potentially be associated with the symptoms. So if a person is having, for example, ADHD, if they're having attention problems, and we know that the frontal lobe is the part of the brain that handles attention, or one of the parts of the brain that handles attention, we might want to do some kind of scan, CT scan, MRI, and look at the functioning in the frontal lobe. Now, we have found connections between some disorders and specific brain areas, although this can be tricky because there is so much overlap in the brain. It is incredibly versatile, and different parts of the brain can sometimes fill in for another part of the brain if it's damaged, that kind of thing. But the example it gives here is Huntington's disease. I don't know how much you know about Huntington's disease. Briefly, it's a genetic disorder, highly genetic. If a parent has Huntington's disease, then all of their children will have a 50% chance of developing Huntington's disease later in life. So Huntington's disease happens usually in around the 40s, maybe 30s to 50s, where a person starts experiencing these symptoms, and the symptoms are often compared with Alzheimer's and Parkinson's and symptoms like that. So a person might have kind of a gradual change in mood, change in maybe cognitive functioning, so some memory loss and cognitive impairment kind of combined with motor skill issues, difficulty with movement, that kind of thing. So Huntington's disease is a very serious disease that we don't have a cure for, and it seems to be associated with the basal ganglia in the forebrain. So this is one example where we have a pretty clear-cut location in the brain that seems to be associated, but we don't always have those locations for all disorders. Oftentimes when we're thinking about the biological model, we're going to be thinking about our brain chemistry. We're going to be thinking about neurotransmitters, which if you have not heard that term, you're about to, so be prepared for that. Information is communicated in the brain in the form of electrical impulses and chemical impulses. So we're going to talk about each one of those in turn. So I'm going to come back to that slide to make sure you guys get a chance to write it all down. Here's what happens. This is a neuron. This is a basic drawing of a neuron. A neuron is a nerve cell. So your nervous system is made up of billions and billions of neurons. And your neurons need to be able to communicate with each other. So when an information comes in, a message, an impulse comes in through the dendrites, that's the location where it comes in. It comes in through the dendrites, which are these little tree-like projections coming off of the cell body. So the impulse comes in through the dendrites, and it's going to be carried down the axon to the end of the neuron, which can be called terminal junction, terminal button. There are many different terms you could use for that. Now, this is an electrical process. The information comes in through the dendrites, and then through a series of electrical processes, it's passed on down the axon to the end of the neuron to the terminal junction. So this is the electrical process that we're talking about here. So we have that written down. An impulse is received by the dendrites of one neuron. It travels down the axon of that neuron, and then it's going to be transmitted through the nerve endings to other neurons. So the first step is electrical. Comes in the dendrites, goes down the axon to the end of the neuron, which we call a terminal junction, terminal button. But what happens next is going to be chemical. Once this message gets here, it needs to pass to the dendrite of the next neuron so it can continue the process. In the dendrite, down the axon to the terminal junction. In the dendrite, down the axon to the terminal junction. Over and over and over again. Here's the problem. The terminal junction or the terminal buttons, the end of the neuron, does not actually touch the dendrite of the next neuron. So it has to pass its message here in a different way. And this is going to be a chemical process here. Neurotransmitters are these little tiny chemicals here that are in these little vesicles that are just waiting to be used. Your brain produces them, and they're just waiting to be used. When the message reaches the end of the neuron, it triggers the release of neurotransmitters. The neurotransmitters cross this gap, which you can see is called a synapse. The neurotransmitters cross the gap, and they bind with the receptors on the dendrites of the next neuron. So it's like a lock and key. We have these keys sitting here waiting. When the electrical impulse reaches them, they are released into this little gap, and they float across, and they go into the lock on the dendrite of the next neuron, and that causes the message to continue. It causes the message to pass. And then after the neurotransmitters have done their job, they're taken back up into the terminal junction to be reused again. That's an important thing to understand, because right there in that synapse is where a lot of our medications do their work. So this is what I've just been saying. Neurons don't actually touch each other. There's a synapse, or a synaptic cleft, or a gap, you'll hear different terms, in between one neuron and the next neuron. So the message has to move across, and it needs to move across in a chemical way. So when the electrical impulse reaches the terminal junction, the end of one neuron, then it stimulates the release of neurotransmitters, which are just sitting around waiting to do their job. They travel across the synaptic space, or the gap, the synapse, to bind to the receptors on the dendrites of the neighboring neurons. So there are specialized receptors for the different types of neurotransmitters. The lock and the key fit each other. Now, there are many different types of neurotransmitters. Some neurotransmitters calm down the neurons. They tell the neurons, hey, stop firing, we don't need to fire anymore. So what happens when you drink alcohol and your brain slows down and things get foggy? Some of you guys may be 21. I'm sure if you're not 21 yet, you have no idea what I'm talking about, right? But your neurotransmitters can also slow your brain down. So drinking alcohol can cause the release of neurotransmitters that slow your brain down. But there are also neurotransmitters that tell your brain to fire. So there are neurotransmitters that increase the rate of firing. So what happens really when you drink caffeine or any other kind of stimulant? What happens? Well, the neurotransmitters that are released are telling your neurons fire more. So you have different kinds of neurotransmitters that do different kinds of jobs, and different kinds of drugs or substances impact those neurotransmitters. So we have identified dozens of neurotransmitters, and there may be some neurotransmitters that we have not yet identified. You've probably heard of some of these. Serotonin, dopamine, GABA are just some examples here of neurotransmitters that have received a lot of research study. GABA is going to be one of the ones that's going to be impacted by things like alcohol that kind of slow your brain down, those inhibitory neurotransmitters that slow down firing. So when we're talking about psychological issues, serotonin is one that we hear about a lot, right? With people who have depression or people who have anxiety, sometimes the theory is that they don't have enough serotonin, which means that we would give someone a medication to increase their level of serotonin, and then hopefully that would be helpful for them. So if you've heard of SSRIs, things like Lexapro and Zoloft and Paxil and others, those medications are, they're SSRIs, which stands for selective serotonin reuptake inhibitors. What that means is that after the serotonin has done its job, it's typically taken back up. But if we give a medication that slows that process down, that leaves the serotonin in the synapse longer, then the serotonin can have more of an effect, or at least that's the idea. So that a person who has depression or anxiety could benefit from having their serotonin stay in their synapse longer. Dopamine has also been connected with potentially anxiety and mood disorders. There are some antidepressants that work on dopamine rather than serotonin. But dopamine is also associated with things like ADHD and schizophrenia. So it could be, from a biological viewpoint, that a person has a psychological problem because they have too much of a particular neurotransmitter or not enough of a particular neurotransmitter. It could also be that there's a genetic concern here. So if we believe in a biological model that your problems are physical, and we know that our physical makeup is impacted greatly by our genetics, then it would make sense for us to find that at least some psychological conditions are genetic. And this is the case. There are certain disorders that seem to be highly genetic. So, very briefly, every cell in your body contains 23 pairs of chromosomes, so 46 chromosomes altogether. You get half from biological mom, half from biological dad. Now, those genetics determine a lot about you. So there's quite a bit about you that is already set at conception. So exactly what you're going to look like, and are you going to be male or female, or are you going to have potentially certain health problems, so that we know that there are physical problems that are genetic. So what about psychological problems? Yes, there is research to say that things like, especially bipolar disorder, which we'll talk about later, schizophrenia, those are two of the ones we see the most, are highly genetic, which means that if you have a close family member that has those disorders, you're at a much higher risk of developing those disorders yourself. Now, it's not 100%, which tells us it's not entirely genetic, but you are at an increased risk for that. But in addition to your genetics, we do also want to take into account your environment, parenting you received, peers, neighborhood you grew up in, those things as well. But there's quite a lot that is set about us and about our temperament and our personality and our mood from our genetics. Also, if we're thinking about things from a biological perspective, evolutionary psychology studies or has the idea of what are the things that are making us more successful at reproduction. Because whatever traits make you more successful at reproduction will then allow those traits to be passed on. So, for example, if a particular person has a trait that makes them attractive, then maybe they're more likely to have multiple children and then pass on those genetics to their children. If there's a particular trait that makes you not able to have children or makes you have health problems that could impact your ability to reproduce, then that would not be passed on as much and would not become as popular in society. So we know about medical conditions. What about psychological conditions? Well, what if there were psychological conditions that in the past helped us to survive? And helping us survive gives us a much better likelihood, obviously, of reproducing. So it has the example here of fear response. Anxiety may have been a really, really good thing back in the day. Now, anxiety is a good thing today to a certain extent. A person who has no anxiety is a person who does dangerous things and will be very likely to be injured or killed. But think back to a time when we didn't have safe homes and safe food and other things. If you were living in a situation where you might be attacked by a predator at any moment, having that startle response and being anxious might have been really helpful for you to help you continue to survive. However, it may not be as adaptive today because we're less likely to be attacked and eaten by a predator, but those kinds of things can still linger. So there's also an idea here that evolutionary psychology could play a role. Now, if you have a biological model, then you're going to emphasize biological treatments. You'll be more likely to want to treat a problem with drugs or electroconvulsive therapy or psychosurgery. Now, these are all three biological treatments, medical treatments, but they are listed here in the most common order. So drug therapy is going to be substantially more likely to be tried than electroconvulsive therapy and psychosurgery. And I would say that electroconvulsive therapy is probably substantially more likely to be tried than psychosurgery. So let's take these one at a time. We talked a little bit about this in our history slides. Drug therapy really began in earnest in the 1950s with psychotropic medications that had some nasty side effects involved. but that were helpful in people being able to return to their typical environment and not be institutionalized anymore. 4 major drug groups. So we have medication intended to treat anxiety, which is primarily of the tranquilizer sort, and we'll talk more about this when we get into anxiety disorders. Antidepressant drugs, or drugs to treat depression. Antibipolar drugs, so drugs to stabilize mood. We'll talk more about these in the different sections. And then antipsychotic drugs would be drugs to try to reduce symptoms of things like, for example, schizophrenia. So trying to reduce things like delusions and hallucinations. All right, so what I want you guys to be thinking about today would be the advantages and the disadvantages of using medication to treat. Specifically here, I ask about anxiety and mood disorders. So when you have a biological model, you're much more likely to choose a medication as a treatment. And there will be some clients who will want this. They'll want to go see a medical doctor and be given a prescription, and that will feel more comfortable for them than going to see a psychologist and doing therapy. What I want you to think about is both sides here. I want you to think about what could be some good things about using medication instead of therapy, what could be some bad things about using medication instead of therapy. So you could think about the cost effectiveness. You could think about side effects. You could think about whether this teaches coping skills or other things that we need to be successful. Maybe what happens if you stop taking the medication. So I want you to think about the good things and the bad things here. I'm looking for your thoughts. You should be able to answer this based on this lecture, based on personal experience, maybe with medications you've taken yourself or medications you've seen other people take. I just want you to think, if you're a psychologist and your client says, well, I'm thinking about taking Zoloft for my OCD, you need to have at least some idea of, okay, well, here are some good things about doing that. Here are some bad things about doing that. Your own personal opinion about whether or not medication is the way to go for treating things like anxiety and mood disorders. So I'm just looking for your personal opinions here. All right, great. So drugs are going to be by far the most common biological treatment today, but there are a couple of other options. Electroconvulsive therapy, or ECT. This involves, doing this today involves giving anesthesia, so the person would be unconscious, and then you would basically induce a seizure. You would give an electroconvulsive shock that would induce a localized seizure in a particular area of the brain. Now, this is kind of contrasted with the way that electroconvulsive therapy began, and something that you might see in different movies. I don't know if you guys have seen One Flew Over the Cuckoo's Nest, but I think anyone interested in psychology should see One Flew Over the Cuckoo's Nest. But there has been a history of electroconvulsive therapy being done in less civilized ways. So initially it might have been done possibly without consent, which would never happen today. It might have been done without anesthesia, might have been done with high levels of voltage, less precision. So electroconvulsive therapy is certainly more refined today than it previously has been. This treatment is considered kind of a last resort for depression. So it's especially helpful for depression. That's where we typically see the best effects. So someone who has depression and goes in seeking treatment, we wouldn't start with electroconvulsive therapy. I'm sure we would start with a medication. But what happens if a person has been attending therapy and taking medication for an extended period of time and they're just not achieving the benefits that they want? Electroconvulsive therapy is an option, and this treatment is used on tens of thousands of depressed persons annually. However, you can imagine that it would be a last resort because in addition to being a pretty serious treatment, it does have side effects. Things like memory loss in particular is one that we see sometimes, but electroconvulsive therapy is still done in some forms for people who have depression. Psychosurgery or neurosurgery is something that is extremely rare today and would only be used in the most extreme cases. As you see here, it has its roots in trephination, as we talked about, drilling holes in the skull to try to reduce symptoms because the person thinks that maybe this will allow the evil spirits to escape. Lobotomy is often what we're hearing when we're talking about psychosurgery. The idea of a lobotomy was to insert some kind of instrument into the brain and to remove a section of the brain or to disrupt the connections between different sections of the brain. And so this could have been achieved in a similar way as trepanation, drilling a hole. There were times when this could have been done by inserting something in through, like, under the eyelid in that area. I know it'll make you cringe thinking about it. Lobotomies were done beginning in the 1930s. There were people who were having no quality of life, who were institutionalized, and this was the new idea about how to help these people. Unfortunately, there were very mixed results. There were some people who did get better, amazingly, after a lobotomy. There were some people who were unchanged, and there were some people who got worse, as you can imagine. So very mixed results. Probably a last-ditch effort. Once again, I do encourage you to watch One Flew Over the Cuckoo's Nest. It's not required. There's not going to be any test questions on that, but if you're interested, this is a great movie for you to see. Today, it very rarely happens and is much more precise. One psychosurgery that could potentially happen today would involve severing the corpus callosum, which would be the connections between the right and left half of the brain. That would only be done in severe situations, but it can be helpful for very significant epilepsy. So psychosurgery is not something that happens much today. All right, last thing to be thinking about when we're talking about the biological model. There are some good things about it. It does enjoy considerable respect. Psychology has often been looked down on, and you can understand why people might be a little suspicious of psychologists. So having a medical model seems to give us more respect in the field. And also, it does produce quite a lot of research. So there are a lot of research studies about brain anatomy, brain chemistry related to psychological problems. And the treatments can be very helpful. Even electroconvulsive therapy sounds very negative, but it can be very helpful for people who have not achieved any benefits from other treatments. But a lot of people have been helped by medications. People have been able to kind of get back to a normal level of functioning. Unfortunately, though, no model is perfect. There's a concern that the biological model is too simplistic. So if you're only looking at your neurotransmitters and you're not looking at the environment, you're not looking at the parenting and the peers and the culture, you could be missing some very important influences there. And then also, these treatments can have negative effects. So obviously, neurosurgery and electroconvulsive therapy, but also drug treatments. Any kind of medication has a risk of side effects, and some of these side effects are pretty substantial still, even though we have worked hard to reduce those. So we have no perfect models, but there's certainly something, even people who have a more psychogenic view of where psychological problems come from would still say that there are certainly situations where the biological model is appropriate. We know that there are genetic contributions to psychological problems. All right, so that's all for this lecture video. If you have any questions, let me know. Don't forget about Activity 2, and I will talk to you guys in the next video.